Hi everyone. It's really great to see so many people here and in the audience and online. And it's a great pleasure today to welcome Alexander Stoessel. Alexander is from the Friedrich Schiller University in Jena in Germany, but he's also closely affiliated with the Department of Archaeogenetics at the Max Planck Institute for the Science of Human History in Jena. And he's an evolutionary biologist. And he's got a great interest in human evolution and functional and evolutionary morphology of vertebrates, particularly focusing on mammals and birds. But his main research interest is really into the function, evolution, and variability of the organs of acoustic communication, which is kind of a nice way of saying ears and the inner ear and so on, right?
Yeah.
And so we're really excited to welcome him today to give his talk. Afterwards, as usual, there's going to be some opportunities for asking questions of anyone in the audience and online. If you are online, you can write a little comment in the chat, and we can then share your question with Alexander at the end. But for the time being, I'd like to hand over to Alexander to give his talk. Thank you so much.
Thank you, Tom, and thank you for this very nice introduction of myself, and also thank you for this very kind and generous invitation here to the University of Vienna and to this beautiful city. I know it from the past. It's so foggy, so unfortunately yesterday I didn't see anything, and I'm very proud to present or to be able to present parts of my research here in front of this very impressive HEAS, I hope I pronounce it right, HEAS network. And myself, I'm also part of a network between the Friedrich Schiller University in Jena. Here you see the world famous Phyletisches Museum founded by Ernst Haeckel, where actually my office is, and also the Max Planck Institute for Evolutionary Anthropology in Leipzig, where I spent 6 years of my postdoc time until 2016. And I'm also well aware that like working on such a large project like the evolution of the human ear needs to a joint investment with many people. And I just want to take the chance to show you or to mention some of my oldest and most closest collaborators without whom this research project would never have been possible. Most importantly, Philipp Gunz. He has also a long history here at the University of Vienna, and Johannes Krause. And I start my presentation with showing this fantastic diorama, which you probably all know, which was shown in the Human Evolution Hall and the Natural History Museum in New York, trying to depict the makers of the famous Laetuli footprints, probably to Australopithecus afarensis. And of course, lots of the details shown here probably are not so correct. Probably it's also very unlikely that they were walking arm in arm like lovers through the open savanna with giant hyenas and lions and all kinds of other stuff present. However, this diorama, this drawing, has really inspired me ever since and really fascinated me. I find it even kind of mystic, to be honest. And there's one specific detail which we see when we zoom in. These two individuals are depicted with their mouths open, and it almost seems like they were like vocalizing or communicating, or were they even talking to each other? These are really fascinating questions. And yeah, I'm asking myself, how were these early ancestors of us like hearing their environment, or when they're communicating to each other? And looking at this and realizing this, I became quite aware a couple of lots of years ago now that we know really few things only about vocal communication or communication in particular hearing in fossil hominins. And these are the questions I'm really fascinated in, and which sometimes even rob me sleep. And I want to present parts of this in the next few minutes. And since when wanting to understand vocal communication and hearing in extinct species, of course, we have to look at parts that become fossilized or that fossilize. And since the vocal apparatus like the larynx and also the brain do not fossilize, it's only the ear, the hearing organ, that actually fossilizes, which I try to convince you is worth looking at. So to show you where the ear is actually at, I show you this chimpanzee skull reconstruction. And when we make it transparent, you see directly here in colors, the ear region, which is made out of, and that's much better seen here in the schematic, made of three phylogenetically, functionally, and also ontogenetically distinct parts. You have most in the periphery, the external ear with the pinna and the external ear canal. Then basically in the middle is the air-filled middle ear, housing the three tiny ear ossicles. Three ear ossicles is a derived feature of all mammals. And finally, you have the fluid-filled inner ear, which is made out of two parts. One part is the vestibular system. I come into detail in a few minutes. And this spiral thing here is called the cochlea, which is the actually receptor organ of hearing. And while the external ear and the middle ear and the cochlea are dedicated to sensing hearing, it's the vestibular part of the inner ear that is involved in sensing spatial awareness and balance. And these two parts will become important in my talk. So here you can see the three main foundations of my research within the past 10 years, I would say. So in basic, I study morphology and function of the ear region in mammals. And one aspect is studying the evolutionary morphology and variation of the ear region, and here in particular of the inner ear part of the bony labyrinth, so the red part here. And I want to convince you in the next few minutes that by studying the variation of the bony labyrinth, we can also trace the dispersal of humans across the globe. And this is particularly interesting in the absence of ancient DNA. I also use experimental approaches to study hearing, and with the focus on the evolution of hominin hearing, which is particularly relevant also in the origin of language. And here the main question is, what are actually great apes hearing? And I will show you that we know surprisingly less about great ape hearing. And finally, we as a group are interested in the function morphology of the soft tissue inner ear, of the membranous inner ear, which is housed inside the bony labyrinth. And this is crucial for studying the function and understanding the function of the hearing organ and the organ of balance. And we use this research to study also the evolution and origins of human bipedal locomotion, or the origins of endothermy, so warm-bloodedness in mammals. And today I mainly focus, I will walk you through the first two research aspects, and the third one, the function morphology of the membranous inner ear, I will only tease at the end of my talk. So let's come to the first aspect. Can we use the bony labyrinth in the absence of ancient DNA? And so this topic would be tracing dispersal, variation, and affinities of Homo sapiens. And here you see a map that broadly shows you the dispersal of humans across the globe during the last 300,000 years. And I'm well aware that many of you working here in Vienna have contributed substantially to this map. And I'm also well aware that the rise of ancient DNA research has really revolutionized our understanding on thinking of the dispersal of modern humans across the globe, of human population history, and also on the affinities of human communities and groups, be it extant or fossil. And however, due to climatic regions, often there are parts of this world where ancient DNA preservation is not present, sometimes is not given, particularly from the tropics and the subtropics. Also, often fossils are way too old for ancient DNA preservation, and often fossils cannot be destructively sampled. So, and this you can see here with these question marks, concerns particular tropical region, as said, and that's why, or therefore, we are looking for ways to trace human population history based on cranial remains, and thereby we also want to contribute more to our understanding of the variation of this of the variability of humans from regions which have been absolutely underrepresented in comparative studies so far. And this mainly concerns sub-Saharan Africa, but also Southeast Asia. And to do so, we need to look for cranial regions that are known to reflect a neutral-like pattern of variation, being relatively less influenced by environmental factors or local adaptation. And the work of Katarina Harvati and Tim Weaver has shown that one part of the cranial base, so the temporal bone, basically shows this neutral-like pattern of variation when doing morphological studies or morphometric studies of a human sample, a worldwide sample. And even more so, or in particular, this is true for a structure or organ that is highly integrated within the temporal bone, which is the bony labyrinth. And that has been shown by Ponce de Leon et al. Already. So, which is the bony labyrinth or the bony encapsulation of the membranous inner ear. And as you see here, this is a human skull, transparent. It is housed deep inside the petrous portion. It's a petrous in German, Felsenbein, a very hard bone. The petrous portion of the temporal bone, and is made out of two functional units. I mentioned it earlier. We have the vestibular system dedicated to sensing balance and spatial awareness, made out of this round-shaped vestibule and the three semicircular canals, which sense rotational accelerations, and the spiral, now maybe I try to be able to say it, snail shell-shaped cochlea, which in all Therian mammals, which are the marsupials and the placental mammals, has this coiled, snail-like shape. And the bony labyrinth is interesting for our research questions from many perspectives. First of all, it is well known. That is, it contains a lot of taxonomic information, as you can see here in this comparison of a bony labyrinth of a Neanderthal and a modern human. And when you are staring like for years onto these bony labyrinths, you would easily recognize that in Neanderthals, this part is relatively shorter than in modern humans. And this canal, which is the lateral canal, sits much higher than in modern humans. Also, as I said, in German, it's called Felsenbein. So the old anatomists, they had a real comparison to bone, to rock-like bone. It's the densest part of your body, apart from the teeth, of course, and therefore it's often very well preserved in the fossil record and with quite a high frequency. So if there is anything left of a hominin or a mammal, then it's teeth or the bony or the petrous bone containing the bony labyrinth. Here you see an australopith. And finally, its development is under very tight genetic control, and nicely, it's fully formed well before birth. So it fully forms around the 25th week of gestation. And this is the nice thing about it, is that's why we can include juveniles into our studies. And we have a strong, as you all know, a strong bias towards juveniles in the fossil record. So that allows us to really increase our sample. And for the first study, this is the sample. The first study I want to present, tracing the dispersal of humans across the globe. And I was able to collect, I would say a very nice sample of Holocene Homo sapiens. And I'm very proud about many collaboration, particularly with sub-Saharan African institutions. And this sample really filled a lot of gaps of the global sample. And then I want to show you, in addition, here for this talk, 10 early Homo sapiens specimens dated between 315 and 100,000 years before present. I agree, and I admit, basically, that this is kind of a wastebasket, early Homo sapiens. But for this study, it is sufficient. From earhood to the laterally hominid 18, the Singa cranium, skull, and Kafze. Then we have, I'll show you 11 upper Paleolithic European Homo sapiens specimens from Sladikun to Apripato and Lagavelio. I added 21 Neanderthals from a very broad temporal range and also a broad geographic range from Crimea to Gibraltar. 7 specimens from the Ibero-Mauritian site, late Pleistocene site of Taforalt. And my favorite fossil actually. Here you see this purple star, a fossil very recently described from Liang Tebo. It's an eastern tropical rainforest of eastern Borneo, dated to 31,000 years before present. And all of this, the individuals I will show you here either come from an archaeological context and that have been CT scanned prior to ancient DNA sampling, or they come from repatriation projects where usage of the microcities was specifically allowed, or they come from collaborations with with local communities and institutions. And I'm well aware of that working with human materials really requires some special ethic responsibilities, and we really try to live up for that. And in this context, I really want to thank the San National Council of South Africa also for their engagement and approval of this research, and similar collaborations we have with lots of other institutions. And yeah, we have in Jena and in Leipzig each one micro-CT lab. You have one here in Vienna as well, or at least one, I guess. And I'm running the one in Jena, and this is our micro-CT, which all our 3D reconstructions are based on that I'm showing you. It's a very nice SkyScan 211 X-ray nanotomograph. And I also, if we are not, actually I want to brag here a little bit, if we are not allowed to bring the fossils to Europe or to Germany, we travel to the fossils. And I actually only want to show you this. This was in 2015, and this dream in my life basically come true, a working dream. There are other important things in life, but from work-wise, when I was able to scan Lucy, so the famous AL288 specimen from Ethiopia, dated to 3.3 million years. So that was really a highlight. And I'm here at the place where 3D geometric morphometrics or 3D shape analysis was actually, yeah, I would say invented. So I don't have to go so much into detail into shape analysis, I guess, or 3D geometric morphometrics. But based on the CT data, we reconstruct the bony labyrinth three-dimensionally, and then we apply a GM, a 3D geometric morphometric or shape analysis approach. And we really try to, or we did a comprehensive landmark protocol, placing a large number of landmarks, up to 250, along the curves of the bony labyrinth and also the central streamlines. And this landmark protocol was designed in a way that it captures all the biological relevant shape of the canals and also of the cochlea. And after that, we do the actual shape analysis, which first is a procrustes superimposition, which basically, you probably all know about it, removes a lot of information from this landmark. So it removes size, it removes spatial orientation, and position in space, orientation and position in space. And then you get shape variables or procrustes variables. And these procrustes variables can be analyzed using multivariate statistics. And in the next slide, I will directly jump into the results. And what you see here is the results of a principal, there are some question marks, which there's a formatting problem. So I'm not doubting that. What you see here is a principal component analysis, or in this case, even a between group principal component analysis of the procrustes shape variables of the coordinates of the bony labyrinth of our approximately 500 specimens. And you see a three-dimensional shape space that is defined by PC1, PC2, and PC3. And this color or grayish things are convex hulls. They represent basically the variation of the specific groups. And what you see here is that the variation of Holocene Homo sapiens is enormous. And it's the entire variation that is very large, but it's also the intra-community or intra-group or intra-population variation that is very large. It's something that is well known for modern humans. And what you also can see is that fossil Homo sapiens, so the early Homo sapiens cluster and the Upper Paleolithic European Homo sapiens cluster, they fall well into the Holocene Homo sapiens variation. And Neanderthals, in contrast, are quite well separated from Homo sapiens, particularly along the axis of principal component 2, and that really implies that their bony labyrinth indeed looks distinctively different. And now you see here these grayish spheres, and these grayish spheres, they represent the means or the mean shapes of the Holocene Homo sapiens groups. And like San and Khoikhoi or Polynesians and so on and so on. So each sphere is one mean of the group. And when adding the names of the groups, it becomes quite easily visible that along the first axis, there is a gradient in Holocene Homo sapiens, and this gradient goes from early Homo sapiens and sub-Saharan African groups on the left side of this plot towards regions far away from Africa, particularly the Americas or also Polynesia. And it was very fascinating to see what this gradient actually is. And this gradient is depicted here. You see a shape, or basically an average shape for early Homo sapiens or sub-Saharan Africans. So basically what you saw on the left, and here the shape for the mean shape for Americas and Polynesia, and this gradient that we saw, this gradient out of Africa, is a reduction in the coiling of the cochlea, and that was to me very surprising, given that it's also functionally a very important organ. However, I directly want to say I don't think that it has any functional relevance. It's probably something related to a random genetic drift or something. So I don't want to have any, I don't want to proclaim any functional interpretation of this result. However, it's an important result still, and I gave a presentation a year ago in front of the African Association for ENT, for otolaryngology, so ENT doctors, and they were completely struck by this result because it has implications for how you place cochlear implants. If you have almost a turn more in your cochlea, then you have to design your cochlear implant differently than if you have just 2 or 2.5 turns, like in many indigenous Americans or indigenous Polynesians. And these differences can become quite dramatic. Here you see the extremes of the real bony labyrinth reconstructions, and it's easily observable how different these cochlea actually are looking. And what you see here next is a plot showing the bootstrap total variances of these shape variables in the groups of which contain more than 10 individuals in our sample. And the more, basically what is shown, the more on the right side, the more variable the bony labyrinth morphology within this group is. And this is a nice plot because it basically shows that bony labyrinth morphology also reflects genetics, because not only do the mean shapes of the bony labyrinth show a gradient out of Africa, but we also see a more or less general gradient from out of Africa in the variances. So with the highest variance we see in the San and Khoikhoi community, and then going from out of Africa, there are some exceptions, I have to admit. Going from out of Africa, the shape variance clearly decreases and is lowest in the indigenous people of the Americas and Polynesia. And this is a phenomenon we know also from genetics. We know that genetic variation decreases when going away from more away from Africa. And we can also add the 3 fossil groups, and early Homo sapiens shows quite a large variation, but this doesn't come as a surprise because, as I said earlier, this is kind of a wastebasket group. It probably comes from the deep time history of this 200,000 years the individuals are coming from, and also from a deep population structure. However, interestingly, Neanderthals are on the lower end of the shape variance, which might be surprising at first because they also come from this broad temporal and also geographic range. However, given the small estimated population sizes of Neanderthals and the high amount of inbreeding, this result probably isn't that surprising anymore. But importantly, for the next few slides, is shape variation of the bony labyrinth kind of reflects genetic variation as well. And if we want to understand more about the evolutionary history of the bony labyrinth and Homo sapiens and anatomically modern humans, we have to look a bit more closely into the fossil groups. And when regarding early Homo sapiens here in red and looking at the average labyrinth shape of early Homo sapiens, then it becomes evident that there are two sub-Saharan African communities. These average shapes of the bony labyrinth are most similar to. So first off, it's the Central African hunter-gatherers, here shown in this blue convex hull, and even more so the San and Khoikhoi, whose average shape is most similar to the early Homo sapiens shape. And this basically allows the conclusion that early Homo sapiens morphology is included in the sub-Saharan African variation. The placing of the Upper Paleolithic Homo sapiens cluster is much more difficult and is for me still unresolved actually, because it's not that easily visible, but they are plotting directly in between the Homo sapiens cluster and the Neanderthal cluster. And the shared feature, or the feature they basically share, is a shared relative enlargement of the lateral semicircular canal. I have to leave you without a definite answer to that, but thinking about it, it could be related to somehow to integration into the cranial base. There might be some similarities in this early Upper Paleolithic Homo sapiens and Neanderthal cranial base morphology, and this might be related to that. I will come back to that in a few slides. And finally, I promised that I want to show you that I'm convinced that we can use bony labyrinth morphology for assigning or connecting fossil specimens in the absence of ancient DNA. And for an example, I brought with me, that sounds weird, I want to show you two examples of two bony labyrinths that have not been analyzed and published before. So first of all, it's the Sladikun individual. Probably you all attended Cosimo Post's talk a few weeks ago. If I saw this right, he probably already talked about Sladikun from a genetic perspective. And it's a skull that has been found in parentheses in a museum in Prague, in the Czech Republic. And Tom probably can sing a lot of songs about it because it's undateable. At least the cranial remains have not. I don't. Maybe you can tell us differently, but the cranial remains were unable to be dated with radiocarbon dating, so it produced a lot of different results. And the reason for that is probably it's full of glue, made of cow bones. And my naive interpretation is that this basically makes it impossible to date the human remains. However, Cosimo and his colleagues were able to extract ancient DNA, and it's a female specimen. It's a very interesting specimen because they were able, or I can even say we, because I'm co-author, we were able to date it genetically based on the Neanderthal segment lengths, which were quite long in the specimen. And it is a specimen that must be at least 45,000 years old. And it belongs to a now basically completely extinct branch that is the oldest known branch splitting off after the out of Africa event. So it's basically the oldest known human skull outside of Africa so far, and to my knowledge. And what you can see here is that Sladikun, and since it has been based only so far on genetic data, there were doubts here and there about its age. However, it plots completely within the Upper Paleolithic cluster. And actually, it's the specimen that is most similar to the Neanderthals. It's a bit hard to, we can do this also mathematically. It's a bit hard to visualize, but you have to believe me, basically. So to me, it's no doubt that this specimen is ancient. And the other specimen is what I promised earlier, is Liang Tebo from Borneo. It's from a burial in a karst cave, eastern Borneo, Indonesian part of Borneo. And it's a very interesting specimen anyways, because it was shown that it got its foot amputated with stone tools. And she survived this amputation at least 8 or 9 years. So it's the oldest successful amputation that we know from the human archaeological record. So they were successful in the Bornean rainforest a long, long time ago with amputations. And even more interesting is that the karst cave, so the Tebo cave itself, but also the surrounding caves, are known to house some of the oldest figurative cave paintings, wall paintings, showing these hand stencils and this local fauna. And I now also plotted Liang Tebo, and you can already see where it plots, because this came really as a surprise to me, because I was expecting it to plot more with maybe with Papuans, or indigenous Australian Aborigines. However, it completely plots within the Upper Paleolithic European cluster. And the most similar specimen indeed in shape space is even Slatikun. I mean, that's pure incidence, but that's very interesting. And it is most similar to the mean of Upper Paleolithics. And giving the archaeological context it comes from, and also the archaeological, similar archaeological record in Western Eurasia, also at the same time popping up these figurative cave paintings of fauna and hand stenciled and so on. So this shared archaeological record, at least in parts, I just want to leave this maybe a little bit more open. It's very provocative, but I find it's, in this case, it's interesting that there's also a phenotypic link between Western Eurasia and the cave painters of Southeast Asia. We can leave this for discussion afterwards, but it was a surprising and very interesting finding. And I now slightly move on to my second topic. And you've seen this slide. And while the shape changes along the first principal component are a clear example of isolation by distance, we are still not really sure, as I said, what the difference between Neanderthals and Upper Paleolithic humans relative to the other Homo sapiens actually is coming from. But as I said, I have a sneaky suspicion this could be related somehow to cranial base morphology, where the petrous bone and the bony labyrinth are housed inside. And we all know that modern humans and Neanderthals independently evolved large brains. Brain morphology affects cranial base morphology. The cranial base in Neanderthals is wider than in modern humans, and the petrous bone and the bony labyrinth, they clearly relate to cranial base shape anatomy, and they basically have to follow the changes that are basically impacted by the brain and the cranial base to these organs. So that would be my guess. And we have already shown that this is the case for another part of the ear, namely the middle ear. And we have shown that middle ear morphology clearly correlates with cranial base anatomy, and that we can distinctively relate it. And you see here, maybe I have to show you, walk you a little bit through the slide. Here you see from lateral inside, basically, from you look onto the ear, and the red thing is the tympanic, would be the tympanic membrane, and you are looking perpendicular onto the tympanic membrane, which is part of the tympanic bone. And here in blue, you see the oval window, and this oval window houses the footplate of the smallest bone of your body, which is the stapes. And this is part of the petrous bone. And you see in humans, when looking from perpendicular, the oval window is centrally relative to the tympanic membrane, whereas in Neanderthals, it's shifted, it's strongly shifted when relative to the tympanic membrane. And this means that the tympanic bone relative to the petrous bone have changed orientation in both groups. And this is also reflected in the anatomy of the tiny ear ossicles. So the 3 ear ossicles, the 3 smallest bones of your body, which is the malleus, the incus, and the stapes. And with a bit of luck and good eyesight, I was able to catch in CT scans of Neanderthals, here you see Pêche de la C1, the tiny ear ossicles still housed inside the middle ear cavity. And then we also did a shape analysis of these ear ossicles. And surprisingly, here you see modern humans, Neanderthals, gorillas, and chimpanzees. And that's the example of the incus. The shape of the incus of Neanderthals is so different from Homo sapiens as chimpanzees are from gorillas or chimpanzees are from humans. So there are really surprising distinct shape differences in the anatomy of the 3 ear ossicles in Neanderthals. However, when doing a very Mickey Mousey simple estimation of the function of the middle ear, we found a completely different result. So you can estimate the pressure gain that is achieved by the middle ear, which is important to overcome the impedance between air and fluid. Sound travels through air, and air has a different impedance, basically how it breaks the fluid part, the gas particles, than fluid. And if you would not have, if vertebrates or if tetrapods would not possess a middle ear, they wouldn't hear anything outside the water. You can test this in your bathtub. If you go underwater, all of a sudden you almost hear nothing from outside anymore, because the sound is reflected at the border of air and liquid. And that's why we need middle ears. And when we estimated the pressure gain that is achieved by the middle ear by a hydraulic ratio between the tympanic membrane and the stapes footplate, and also the lever lengths of the ear ossicles, we see that humans and Neanderthals are completely similar. So there are no functional difference in the middle ear despite these profound differences in morphology. And this is completely different when looking at the great apes. So the question was now, can we move this on to something not so Mickey Mousey like here? And I come to my basically last topic, which deals with, do humans actually hear differently than chimpanzees and other great apes? And particularly, do the humans hear differently in the frequency range of spoken language, which is between 125 and 8,000 hertz? And this is very relevant if you want to learn more about fossil, or if you want to estimate fossil hominin hearing, because we have complete ears from all fossil, at least starting from Australopiths, from all fossil hominins. We have very complete ears, particularly here from, it's the Kika child, it's an Australopithecus afarensis. And so we actually have everything in our hands to model hearing in fossil hominins. And can we learn about maybe also the origin of language, which is, of course, one of the holy grails of science, of biology, or of evolution, anthropology, or name it. And it's, of course, the first approach you have to take is to look at our closest living relatives, which you all know are the chimpanzees. And we know from great ape genetics that there are many differences, or there's a lot of differences in the genes which are known to be related to audition, particularly to the hair cells in the inner ear. And this clearly points towards an accelerated adaptive evolution regarding hearing in hominids, so in great apes, including humans. However, of the 28 hominoid species that are extant, we have only hearing data. These hearing data are so-called audiograms. I explain you what an audiogram is on the next slide. These hearing data exist only for humans, millions of data, and chimpanzees. So actually, we don't know anything about great ape hearing. And if you are able to publish bonobo or orangutan or gorilla hearing data, that's probably would be very high impactish. I promise. Almost promise. Shouldn't promise anything. The problem is the chimpanzee hearing data are problematic in itself. So there exists data from two studies. One is from Elder from 1934, that was military research, and it's actually the first audiogram of a non-human primate that we know. So it's also very interesting from historical point of view. And the other is, so this includes 3 chimpanzees, and the other studies from 1990 from Japan, from Kojima, who analyzed 2 chimpanzees. And the problem is, in the frequency range of spoken language, so between 125 and 8,000 hertz, these 2 audiograms or the 2 data or the 2 studies, they completely contradict each other. Either, in the case of Elder, it's depicted in a very bizarre way. I don't have the time to explain it here in this one in detail. However, in chimpanzees are more sensitive to the frequencies of spoken language than humans. And the contrary is coming up in Kojima, and you have to see this flipped. So this is an audiogram, and on the x-axis you see the hearing frequency, and on the y-axis the hearing threshold. Basically, the hearing sensitivity is plotted as sound pressure level given in decibels. And here, the lower you are, the better your hearing sensitivity in simple words. And what you see is here, it's completely, so it's flipped to relative to Elder, but what you see is here, the human curve is constantly up until 8 kilohertz, lower, meaning they would hear better these frequencies than chimpanzees. Interestingly, both studies agree in one aspect, that there is in chimpanzees a dip, a reduction in hearing sensitivity, approximately between 3 and 5 kilohertz, and that is also the range where humans are known to be most sensitive. And we have to solve this contradictory problem first before we can go on. And this is really relevant. You can see here when we try to model average hearing sensitivity in the frequency range of the approximate frequency range of spoken language, and we try to model the evolution of the 28 primate species where we have hearing data. And what you see here, when we use the chimpanzee data based on the 1934 study, a Brownian evolution model with a constant evolutionary rate best explains the data, meaning also that when looking at the last common ancestor of humans and chimpanzees, there actually hasn't happened a lot, meaning that chimps and humans basically have not changed from the last common ancestor, that they are basically hearing the same. It's completely different when looking at data from Kojima. Here, a Brownian evolution model with a change in evolutionary rate from the last common ancestor of human chimpanzees towards Homo sapiens. Quite a dramatic change occurred. And here, this implies really that hearing in humans and chimpanzees is different, and that something happened on the line towards humans. And this, of course, is very tempting to interpret that this has something to do with the origin of language and language per se. So the most straightforward way to do that now would be to get new hearing data from chimpanzees and other great apes. However, at least in the Western world, I would say, but probably everywhere, it's impossible, because obtaining new hearing data would be in vivo, would be invasive research, because it means you have to stick in very, very tiny, it's actually almost not invasive, but it would be invasive, very, very tiny needles onto the skull to measure the auditory brainstem responses. So getting new audiological data from great apes, I would say, is almost impossible. So we have to find an indirect way to measure that. And now it was really worth to look again at the morphology of the external and the middle ear, so what I call the auditory periphery. And external and middle ears, the function in vertebrates is to collect, transfer, and amplify sound. And shown by the work of John Rosowski, thereby the structures, and that's important, they strongly affect the hearing sensitivity. They basically pattern the hearing sensitivity. And they affect hearing sensitivity through a function, and this function can be measured experimentally. It's the auditory periphery transfer function, and it basically gives you the magnitude how sound is transferred through the external and middle ear towards the inner ear. So this way. And when doing the comparative morphology of the auditory periphery, some clear differences between humans and chimpanzees become very easily visible. I can also give you afterwards the hard numbers, but it looks nicer without a lot of hard numbers. And what you see is this, despite a larger average body mass, humans have a smaller tympanic membrane than chimpanzees, but they have a larger stapes footplate and much heavier ear ossicles. So they are distinctively significantly heavier. And even more pronounced are differences in the external ear canal. Here you see digital casts of the external ear canal. This would be where the tympanic membrane would sit, and that's basically here, the outside of the bony part. And what you see here is that humans have a much shorter external ear canal, but a much wider external ear canal. So these morphological differences, they actually should mean something, we thought, and we think. So we decided to test this experimentally. And what we used two approaches to measure sound transfer. In the middle ear, we used laser Doppler vibrometry, which needed us to open up the middle ear surgically. You now think that's highly invasive, but this was done, of course, with cadaveric specimens, freshly dead cadaveric specimens. And here, using a laser that is pointed toward the stapes footplate, you can measure stapes vibrations. And these stapes vibrations come from the excitement of the middle ear. A sound source is plugged into the external ear canal and then excited from 125 towards 10,000 hertz. And then by the displacement of the stapes footplate and the laser Doppler effect, you can actually measure first the velocity of vibrations of the ossicle chain, and then you can integrate it into a magnitude. And for the external ear canal, we used a different approach. We used the finite element analysis approach. And here we simulated the pressure gain that is achieved by the external ear canal. So it's a different approach. And afterwards, we combined the two. I will show you in a second. That was our sample. We had 3 bonobos, 4 chimpanzees, and 12 humans. And you would now say this is a small sample, but what you need fresh or at least very well frozen specimens. And it took us forever to get the sample of chimpanzees and bonobos, more than 5 years actually. So these apes, they don't die that often, and often they are directly put into a fixation, and then you cannot use them anymore. And this is how the theater looks like. I have done this research with the ENT department of the University Hospital in Dresden in Germany, and you need an ENT surgeon to get access to the stapes footplate, and it took 5 to 10 hours to basically open up the tiny middle ear and to carefully remove the mastoid bone and then to resect the facial nerve canal. This ENT surgeon is really to me, an artist, a superhero artist. And then the measurements actually take like 5 seconds. So that's then very fast. So let's come to the results. I torture you a little bit with, at first, complicated looking graphs. Here you see the middle ear transfer function. So how well sound is transferred through the middle ear. These are mean curves. On the left, you see the 2 individual chimpanzee species. So this is green bonobos, red chimpanzees, blue humans, average curves. And here, which probably makes it simpler or easier to observe, there's an average of all the canines, so chimpanzees and bonobos together, and humans. And what you see is that across the entire frequency range, the middle ear of chimpanzees and bonobos transfers sound better than humans. So the higher you are, the better, the greater the magnitude. And it clearly becomes visible that humans and chimpanzees in the frequency range of spoken language, their middle ears transfer sound better than the human middle ear. And we can directly relate this functionally. This is clearly related, particularly here in the frequency range below 1000 hertz, to the hydraulic ratio between the tympanic membrane and the stapes footplate. You see that the ratio is much smaller in humans. And also it relates to the large size of the tympanic membrane in humans and chimpanzees. You basically can see this box plot, this trend here directly in the measured values. These are the simulated pressure gains of the external ear canal, and what you can hopefully easily observe is that blue again humans, that the short and wide external ear canal of humans causes a resonance frequency that kicks in, or that hits in much later at a higher frequency than in the chimpanzees and in the bonobos. So in chimpanzees and bonobos, you have an amplification of sound, a very strong amplification of sound by the external ear at around 3000 hertz or a little bit below, and in humans at 4000 hertz. Interestingly, that's the same frequency range where you have your best hearing sensitivity. Last torture slide, sorry, but that is now the combined middle ear and external ear transfer function. So the auditory periphery transfer function. I quickly walk you through the slide. Again, you have frequencies, the frequencies here, and you have the magnitude of the transfer function, so how well sound is transferred. And now I flipped to confuse you. I flipped again the y-axis, no, not to confuse you. I did this here for this talk in order to make it better comparable with audiograms. So in here, the lower you are, the better sound is transferred through your auditory periphery. And again, blue humans, red chimpanzees, green bonobos, and the orange one is the Panin curve, the average Panin curve. And again, even in the combined transfer function, you see that the two great ape species have a constantly and also significantly greater magnitude of sound transfer than humans, except for a very small bandwidth basically, and this bandwidth is around 4000 hertz. It is where the resonance frequency of the external ear canal is in humans, and it is where humans are best in hearing, and where also the dip in hearing sensitivity was in both the historic studies I showed you by Elder and Kojima. However, the take-home message here is that chimpanzees and bonobos are better in sound transfer through the external middle ear than humans. And this I can come to a conclusion here that again, chimpanzees and bonobos have a better sound transfer than humans in the frequency range of spoken language. That was surprising. And as I showed you, the sound transfer functionally strongly affects hearing sensitivity. It basically paves the way for the hearing sensitivity. And that allowed now the conclusion that actually the data from Elder, which are almost 100 years old, which are the first hearing data from a non-human primate, are actually the correct data for chimpanzees. And this has strong implications, because it means that the ability to capture speech frequencies accurately was already present in the last common ancestor of humans and chimpanzees. So it was already present well before the evolution of spoken language. So it basically has nothing to do with spoken language. And unfortunately, that's why it's probably only published in scientific reports, it's a negative result. We cannot use the morphology of the external middle ear to trace the origin of human spoken language. So sorry, but that's what it is. And it's probably much more related. And humans, indeed, humans are unique in the anatomy of their external and middle ear. I showed you this. And the peculiar or special human ear likely emerged as a byproduct of the evolution of the human cranial base. Again, that's the theme throughout my entire lecture here, is that cranial base integration into cranial base really is the main driver for the anatomy of the ear. So it's a byproduct of the evolution of human cranial base through brain expansion or a shift to bipedalism. And the human brain really expanded laterally, and thereby it decreased the tympanic bone in length, and the tympanic bone after birth basically forms your external ear canal. So your external ear canal shortens, and that clearly has implications also functionally. And importantly, spoken language likely evolved in this context And the speech reception threshold, so sensitivity to speech reception, constrained human auditory thresholds or constrained the hearing sensitivity and not the contrary. So I have 2 more slides left, and I promised a teaser because I just want to quickly show you that the inner ear is, of course, much more than bone. And the actually sensory organs are housed inside this bony encapsulation. And during my time in Leipzig, together with my colleagues, we developed a method in freshly dead specimens to visualize the thin membranes using phosphatidoglycic acid staining and to visualize the thin membranes of the inner ear. And that was really a breakthrough because these membranes, they decay quickly after death, around 24 hours after death. And the Max Planck in Leipzig is directly next to the anatomy building of the university hospital and also to the veterinary pathology. And during my 6 years in Leipzig, I dissected fresh dead mammals and all kinds of from elephants to tigers to all kinds of great apes and also non-mammalian species like a 6-meter long crocodile and everything. It was really a cool time actually, and also fresh human specimens, which made me had to drink some schnapps afterwards because it was it's something different than going to the autopsy room with fixated specimens. So kind of a horror story at the end. So here you see the membranous organ. And this membranous organ is filled with a fluid called endolymph, and this endolymph has a specific viscosity, and this viscosity is important for function. And the lower the viscosity, or saying giving the higher the body temperature of an animal, is the lower the viscosity, the more decreased is the response speed here in these canals. So the response speed of the duct system, yeah, the viscosity, if it's really fluid like water or more like jelly, that really makes a difference. And since these organs monitor head rotations, this has a temperature, body temperature, and viscosity has an impact onto its function. And just as the last slide, led by Ricardo Araujo and Romain David, and studying the membranous labyrinths of more than 50 vertebrates and more than 350 bony labyrinths, including a lot of mammalian ancestors, we were actually able to show, based on the measurements we did on the membranous labyrinths and giving changes in viscosity, and mammals increased their body temperature during their evolution and also are extreme high performers regarding locomotion. So there is a problem relative to each other. And this increase in body temperature basically caused an increase in the viscosity of the endolymph. And this had to be basically, made up by changes in the morphology of the duct system and also in the morphology of the canal system. And we were able to show that there came a sudden and abrupt change in the duct and the canal morphology as a response to increased body temperature, given that they are now warm-bloodedness. And the response was that mammals developed more thin cross-sections of the ducts and of the canals and a relatively smaller radii of curvature. And this morphology suddenly really appeared around, we can date this to 233 million years before present, to the end of the Triassic, to the late Triassic. And this change appeared much later than, for example, parasagittal movements or the development of I don't think. I don't get the English word now. So other structures that are related to, have been related to endothermy. And our conclusion was that endothermy developed much later in mammalian evolution than was previously thought. And my last word would be actually, if you want to study the function of the inner ear, you really have to look at the membranous of the soft tissue morphology first. And just afterwards, you can try to, using phylogenetic informed regressions, you can look at the bony morphology. By just looking at bony labyrinths and predicting some kind of locomotion behavior or what you want, you are very often very wrong. So first study the soft tissue, and then you can look at fossils. So with this, I want to thank you. Since I have so different topics, I don't have concluding remarks here. But thank you very much again for the generous invitation here to Vienna, and I'm happy to answer questions.